terms not required, like this you can remember. Right? And then, so these are the bondings that we have encountered, right? For maltose, lactose, sucrose. Nowhere you are going to get, but you should have an idea. Here it has what? 1,4 glycosidic bond in maltose. Lactose also it is 1,4 glycosidic bond. Here also it is 1,4 glycosidic bond. Sucrose, maltose, lactose has what glycosidic bond? 1,4 glycosidic bond. Next, polysaccharides, they are formed by glycosidic bond only. Wherever glycosidic bond is forming, there hydrolysis of water is taking place, right? And here if we see, we have two types of polysaccharides, homopolysaccharides and heteropolysaccharides. Homopolysaccharides will have only one type of monomer. Heteropolysaccharides will have different types of monomers. Likewise, for heteropolysaccharides, these examples you have to remember mandatory, peptidoglycan and agar. These are not new terms to you, right? Peptidoglycan, where do you see? In the cell wall of bacteria. Agar, it is a hydrocolloid produced by what type of algae? Red algae. Not new terms, right? So, remember this under what? Heteropolysaccharides. Next, homopolysaccharides include what? Starch, chitin, glycogen, cellulose. Here itself, let us try to recall whatever we have. Starch is the storage food material in plant cells. Glycogen is the storage food material in animal cells. Chitin you can see in the exoskeleton of arthropods. It is the cell wall of, it is a composition in cell wall of fungi. Cellulose you can see in abundant composition in plant cell wall. Identified like wherever you can see this, this information is already known to us, right? Now let us look on the add-on information which is given in this chapter. Cellulose has only one monosaccharide, that is what? Glucose. Okay? And what are the bonds present here between glucose unit? Beta 1,4 glycosidic bonds. Okay? And then this is the direct NCERT liner where you have to remember it mandatory. Cellulose is abundantly present where? In plant cell walls. Paper made from plant pulp and cotton fibers are what? Cellulosic. Paper which is made from plant pulp and cotton fibers are cellulosic. And as cellulose has just one monomer and there is no complex formation between that, it just has what? 1,4 glycosidic bond. It will not form any complex with iodine like how starch is forming. Like how starch is forming, cellulose will not form any complex with what ma? Iodine. Okay. So, these are the points you have to remember under cellulose. Simple, it has only one monosaccharide, glucose, with what bond? Alpha 1,4 glycosidic bond. It is abundantly present in plant pulp and used in paper industry, and it is not forming any complexes with iodine, that is all, because it does not have any branched or complexes, right. So, that is it about cellulose. And then starch. Starch has dextroglucose, alpha D glucose units, and it has two types of polysaccharide molecules, amylose and amylopectin. Both of them have glucose only, but the type of branch formation is different. Like amylose have what bonds? Alpha 1,4 glycosidic bonds, and here it has what? Alpha 1,6 glycosidic bonds in amylopectin. Alpha 1,4 and alpha 1,6, both it has. Whereas in amylose it just has alpha 1,4 glycosidic bond. And 80% of starch is of what? Amylopectin. This forms complex with iodine, giving what color? Blue-black color or blue color, that we have learned from childhood. So, cellulose information is clear. Starch also clear, right? It has what glucose units? Alpha D glucose units. It has what? Two forms, amylose and amylopectin. Amylose has just alpha 1,4 glycosidic bonds. Amylopectin along with 1,4 it has 1,6 glycosidic bonds as well, and this forms complexes with iodine. 80% of it has amylose or amylopectin? Amylopectin. That is it. Clear with starch, right? And then glycogen, it has both reducing and non-reducing ends. We have learnt it with the diagram. Reducing end is towards which side? Left end. Left end is reducing end and right end is non-reducing end. this in this diagram. Here you can see this is left end, this is right end. Left end is reducing end because it has free aldehyde and aldehyde groups. Right end, it doesn't have any free bonds, it is already connected, so it is what? Non-reducing end, right? Clear, no? And what type of bonds you can see in glycogen? Alpha 1,4 and alpha 1,6 glycosidic bonds. Clear with glycogen, ma, everybody, right? So what we have learned? Cellulose, starch, glycogen. Now chitin, seen in exoskeleton of arthropods, cell walls of fungi, and it has what units? N-acetyl glucosamine units, NAG units we will call it as. And it is also known as what? Fungal cellulose, as it is present in the cell wall of fungi, it is also known as fungal cellulose. What bond it has? 1,4 glycosidic bonds, that's all, right? And inulin is a polymer of what? Fructose. Inulin, is it absorbed by a human system? No, it will be cleared out. So we can find out the glomerular filtration rate, which is equal to what? Inulin clearance. Clear with carbohydrates? We have learnt everything, no? Revised everything, no? Now proteins. Can you see anywhere homopolymer of proteins? No, because it is built up of what? Amino acids. We have 20 different types of amino acids. Classified, firstly, we have seen different classifications. First, on polarity, we have hydrophilic polar amino acids, hydrophobic non-polar amino acids. And a single amino acid has one alpha carbon along with carboxyl group, amino group and H, which is common for all amino acids. The difference is with R group, which is variable, okay? And first, hydrophilic polar amino acids we will see. Negatively charged aspartic acid and glutamic acid. So these are acidic, right? And positively charged includes all this, of which most noted, you know all this, lysine, arginine, histidine, you already know this. This you have to remember, beta. Serine, threonine, asparagine, glutamine and cysteine, right? And then hydrophobic non-polar amino acids include aliphatic side chain, aromatic side chain. Aliphatic includes what? Glycine, alanine, valine, leucine, isoleucine, proline, methionine. Aromatic, just three you can remember: phenylalanine, tryptophan, tyrosine. So here your memory goes easy with what? Negatively charged polar amino acids, non-polar aromatic side chain amino acids you can easily remember. And this also you can easily remember, like positively charged polar you can remember. But it is little difficult to identify or split between this aliphatic chain amino acids and positively charged amino acids. If you revise it 1 or 2 times, you can remember this also, right? Clear till here? Yeah. And then, yeah. So here you see H group is, R group is just having hydrogen. So this is what? Glycine. If it has methyl group, it is alanine. If it is methanol, it is serine. These are the 3 which are given in our NCERT. Just remember these structures, and it is important to remember others also, but first be thorough with this, okay? And next criteria of classification based on essentiality, whether we are consuming it or not. Those are what? Essential and non-essential amino acids. So just to remember essential amino acids, private Tim Hall, you remember this mnemonic enough. Private Tim Hall. This is phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, lysine, right? So just remember this on the essential elements. Rest out you can easily identify at non-essential criteria. So clear with this, no? And then we have learnt on zwitterion. So I asked one question, where do you see zwitterion in? Neutral solutions, that's all. Amino acid in a neutral solution, it has both positive and negative charge. So any amino acid, it has both positive and negative charge. So it is known as what? Zwitterion. And its value will be what? Net charge will be 0. Okay. And if the pH of the solution is reduced from the neutral solution, if the pH is reduced, then it will be what? +1 of the charge, and it turns to what? A cation. Decrease the pH, so it turns into what solution? Acidic solution. And it is changing to a cation where the net charge will be +1. If you are increasing the pH, then the solution gets basic, and this turns out to be an anion where the net charge is -1. So, neutral solution, acidic solution, this turns to a cation, net charge is +1. Increase the pH, this turns to a basic solution and the amino acid gets a negative charge, it turns to an anion. That is all. Remembered with zwitterion? In simple terms, a zwitterion is a molecule with functional groups of which at least one has a positive and one has a negative electric charge. So, the net charge will be 0, right. On zwitterion also we have seen like 1 or 2 PYQs of your NEET, okay. And then, yeah, peptide bond is formed between what terminal and what terminal? First amino acid carboxyl terminal with amino group of the second amino acid. So, the peptide bond is formed between carboxyl group and amino group. Likewise, for the first amino acid, which group will be free? Amino group. So, this is known as what? N terminal. First amino acid will have what terminal? N terminal. Last amino acid will have C terminal. Carboxyl group will be free. At the center, carboxyl group and amino group are bonded by peptide bond. And we have 4 structures. Now, we are going to discuss about enzymes, right? Most of all the enzymes are of what nature? Proteinaceous in nature. Of course, we have some exceptions, we will discuss that. But bulk, all the amino acids are of what? Proteinaceous in nature, right? And proteins have what structures? Primary, secondary, tertiary and quaternary. Most of the enzymes will have tertiary structures. Their activity or their active sites, when enzymes are functionally active, they are of what structure? Tertiary structure. Primary structure is just like a line, okay. They are binded with peptide bonds. You will have an N terminal and a C terminal, and that just gives what information? Positional information of amino acids. Secondary structures are of 2 types. What are they? Alpha helix and beta pleated sheets. Here also you have peptide bond, but along with peptide bond you have a hydrogen bond, right? No, primary structure is clear to everybody, right? Secondary structure you have alpha helix, where peptide bond is formed between carboxyl group and amino group of 4 down chain. Like that, an helix is formed. Mostly it is what handed? Right-handed helix, right? In beta pleated sheets you have the peptide bond, along with that hydrogen bond is also formed, right? This is of beta pleated sheets. Secondary structure is clear to all, right? And now comes the tertiary structure. Tertiary structure is like hollow woolen structure like this, like a ball structure which is woolen and thread like coiled, and here it has what types of bonds? That is important. Hydrogen bond, ionic bond, dipole-dipole interactions, hydrophobic interactions. Okay? Remember the bonds and tertiary structure of the protein. In quaternary structures you have all the interactions, like alpha subunits and beta subunits. Best example for quaternary structure to learn is what? Hemoglobin, given in our chapter. It has 2 alpha subunits, 2 beta subunits, and this is how you can see the 3-dimensional model or the structure as. Can you see the chains, helix and beta pleated sheets? All are observed, right? So, this is about quaternary structure. All the 4 structures clear, right? And this is the table in our NCERT. After secondary metabolites, the next table we'll see is with what? Proteins, where we'll have collagen, intercellular ground substance, trypsin, insulin, antibody, receptor, GLUT4, glucose transporter. So, all these are what? Proteins. Protein can be a transporter, carrier proteins, channel proteins. Protein can be a hormone, it can be an antibody, okay, it can be a enzyme, okay. So, all these are what? Diversified molecules in the body which are proteinaceous, right? About protein it is clear, no? Next is what? Nucleic acid. Nucleic acid is addressed as what? Like amino acids are building blocks of proteins. Carbon atoms which are binded like monosaccharides, di-oligosaccharides, that will form what? Chain of polysaccharides. Single monomer to homopoly, different monomers to heteropolysaccharides. And then we have what? Nucleic acids. Nucleic acids are what? Polynucleotide chain. It is a polynucleotide chain, wherein single nucleotide if I have to take, it has three components. What are they? Nitrogen base, sugar, phosphate group. If it is DNA, what type of sugar you will have? Deoxyribose sugar. If it is RNA, ribose sugar. If it is DNA, what type of nitrogen bases you will see? Adenine, guanine, cytosine, thymine. If it is RNA chain, what type of nitrogen bases you will see? Adenine, guanine, cytosine, uracil. So the purines will be common, pyrimidines will be changing, right? And phosphate group is common to all. Now comes the bonding. If it is clear to you here, then aram se you can go through mol bio. Okay. So you have 5 carbons. It is a pentose sugar, right? Ribose or deoxyribose. If at second carbon oxygen group is not there, then it is what? Deoxyribose. If this is there, so it is ribose sugar. If oxygen group is absent at second carbon, then it is deoxyribose. First carbon is binded to what? Nitrogen base. First carbon is binded to nitrogen base. Fifth carbon is binded to phosphate group. This simple thing you remember. Nucleotide has 3 components. First carbon binded to nitrogen base. Fifth carbon is binded to phosphate group. First carbon binded to nitrogen base by what bond? Glycosidic bond. Fifth carbon binded to phosphate group by what bond? Phosphoester bond. Clear? Easy? Now. So, if it is purine, nitrogen base is purine. First carbon is binded to what nitrogen? N9 of purine. If it is pyrimidine, C1 is binded to N1 of pyrimidine by N-glycosidic bond, right? And phosphate group is from phosphoric acid. This is binded by phosphoester bond. Clear till here? And these are what? Purines, which are heterocyclic pyrimidines. This table is very important. Might be not asked in the biomolecules, but this will be asked in mol bio. It is on safe end to learn about the IUPAC names of the nitrogen bases. Adenine is also known as 6-amino-purine, guanine 2-amino-6-oxy-purine, cytosine 4-amino-2-oxy-pyrimidine, thymine 5-methyl-2,4-dioxy-pyrimidine, uracil 2,4-dioxy-pyrimidine, right? So this is about nucleic acids, and this is the terminology we have learnt. If it is nucleoside and nucleotides. Just to go through this for a minute, and then we will start on the next one. Any doubt? Done? Done, no? This is what we have discussed till now. And how are 2 nucleotides are formed? What are the bonds involved in a single nucleotide? We have learned. But 2 nucleotides are connected by phosphodiester bond, by 5' phosphate group, and here 3' OH group, right? So in a single nucleotide you have 2 bonds, N-glycosidic bond and phosphoester bond. In dinucleotide or a single stranded DNA or single stranded RNA, you have 3 bonds. What are they? N-glycosidic bond, phosphoester bond, phosphodiester bond. In a double stranded DNA, one more bond is added. What is that? Hydrogen bond, right? Recalled everything? And these are the silent features. Like in a double stranded DNA, this goes anti-parallel to each other. One runs from 5' to 3', other 3 to 5. They are complementary to each other. Number of purines will be equal to number of pyrimidines. And in a double stranded DNA, here if you see, this is known as one complete turn. Is also known as what? Pitch of the DNA, whose length is 3.4 nanometers or 34 angstroms. As it is the stable form of DNA, B-type DNA, it has approximately how many base pairs? 10 base pairs. Distance between two base pairs is 0.34 nanometers and 3.4 angstroms. Clear now? Then, so we have three forms of DNA, A-DNA, B-DNA and Z-DNA. A-DNA is right-handed, B also right-handed, only Z-DNA is left-handed. This you will see in most common in all the cells. And next point you have to remember is number of base pairs. It is 10 in B form DNA, 11 and 12 in A and Z DNAs. Then what? Major and minor grooves. Major groove, narrow and deep. Here minor groove is narrow and deep in B type DNA. Minor groove, wide and shallow, wide and deep. Like that you remember what? A and B type DNA. Right? Z type DNA, major groove is completely absent. Can you see here? No major grooves. Only minor groove you can see, that is narrow and deep. This points you remember enough from forms of DNA. Number of base pairs, major and minor grooves, what-handed helix. 4 points. Right? Clear all? Chargaff's rule. And we have learnt 3 types of RNAs. What are they? mRNA, tRNA, rRNA. mRNA synthesized by what polymerase? RNA polymerase 2. rRNA by what polymerase? RNA polymerase 1. What is the site of the synthesis? Nucleolus. tRNA synthesized in nucleoplasm by what polymerase? RNA polymerase 3. Right? Next, now we have entered with lipids. So lipids we have learnt already, those are water soluble or water insoluble? Water insoluble. But then they are soluble in organic solvents. Do you remember any organic solvents like organic solvents? Benzene, either acetone, all these are what? Organic solvents, right? So, these are the organic solvents into which lipids are soluble, and they could be simple fatty acids or they can be glycerol. Okay, lipids are, they can just have glycerol or they can have fatty acids, and a fatty acid will have what? Carboxyl group and R group. The general formula of fatty acid can be, the general formula of fatty acid can be CnH2nO2. Okay, it has what? Carboxyl group and an R group. Like this. The R group can be anything. It can be methyl, ethyl, or any higher number of carbons. It can range from 19 to 20. Palmitic acid has how many carbons? 16 carbons, sorry. It has 16 carbons including carboxyl carbon. Arachidonic acid has how many carbons? 20 carbons including carboxyl carbon. Right? So what is the intro of lipids like? Lipids are insoluble in water. They are soluble in organic solvents. I have named 3, but the list is wide. You can, if you want, you can remember. And it can just have a fatty acid or it can have glycerol. Those are considered as what? Lipids. Steroids are also what? Lipids. Fatty acid, if we have to take general formula, CnH2nON, the composition can have a functional group with a carboxyl group like RCOH. The R group can be of single carbon ranging methyl or ethyl or higher number of carbons. If we have to take palmitic acid along with carboxyl carbon, palmitic acid has how many? 16 carbons. Arachidonic acid also, along with carboxyl carbons, it has what? 20 carbon atoms. Intro is clear, right, with respect to lipids. Now we will move on to the classification of lipids. Lipids are classified into 3 types. Lipids are classified into 3 types. It can be simple lipids, can be conjugated lipids, it can be derived lipids. Okay. Yeah, listen carefully. So, lipids are of what types? Simple lipids, conjugated lipids, derived lipids. Simple lipids can be true fats, it can be waxes, don't take all this, not required, it's for your idea only, or it can be cutin. These are some types of simple lipids. Conjugated lipids, we have phosphated, chromolipids, we have lipoproteins, all these are what? Conjugated lipids. So, this conjugated lipids are nothing but they will have fatty acid, they will have glycerol, along with that some other components are added. It can be phosphate group, or it can be any other lipid constituents, it can be any other pigments added to it. So, those are what? Conjugated lipids. Derived lipids can have fatty acid, it can have glycerol, okay, but it cannot have that also, okay. It can have or it cannot have, but this exhibits fat-like properties. It will maintain the fluidity. For example, cholesterol. So, the basic classification you have to remember is simple lipids, conjugated lipids, derived lipids. Simple lipid types not required. Conjugated lipids you only remember along with fatty acid, glycerol, some other components are added. It can be phosphate, it can be proteins, it can be pigments. Okay. Derived lipids, it can or cannot have fatty acid and glycerol, but then it exhibits fat-like properties. Got it? Clear all? Yeah. Now, look into the theory once about neutral fats from simple lipids. These are esters of fatty acids and glycerol. They have both what? Fatty acids and glycerol, which are esterified. Fatty acids and glycerol. Glycerol will have 3 carbon components with OH group. This is what? 1 glycerol, also addressed as trihydroxypropane. This is 1 glycerol unit. If 1 glycerol unit is attached with 1 fatty acid, monoacyl glycerol, 2 fatty acids, diacyl glycerol, 3 fatty acids, triacyl glycerol. Okay. So, this is about types of lipids and neutral fats first, okay, which have what? Fatty acids and glycerol. 1 glycerol unit has 3 carbon components, which is trihydroxypropane. Here we go. Before knowing types of fatty acids, you see 1 glycerol component with how many fatty acids? 3 fatty acids, which is what? Triglycerides. Triglycerides. If just 2 fatty acids, diglycerides. 1 fatty acid, monoglycerides. Please correct the term. Okay. Mono, di, triglycerides. Now, when it comes to fatty acids, we have 2 types of fatty acids. You have seen 3 fatty acids in triglycerides. This fatty acids can be what? Saturated or unsaturated. Saturated fatty acids will not have double bond. Unsaturated fatty acids will have double bond. Simple thing to remember. But here you have to remember what? Examples. Examples of saturated fatty acids include what? Palmitic acid and stearic acid. Palmitic acid anyhow given in your book, right? Clear with saturated fatty acids? Unsaturated fatty acids will have double bond between 2 carbon atoms. Example you see, oleic acid and linoleic acid. Oleic acid and linoleic acid, that is palmitic acid and stearic acid. Under saturated, under unsaturated, oleic acid, linoleic acid. Clear na till here? These unsaturated fatty acids can be esterified with glycerol, can turn forming up what? monoglycerides, diglycerides and triglycerides. This one, Ra? Here arachidonic acid, yeah, it has 20 carbon atoms including carboxyl carbon. It is under saturated. We take it under saturated. Okay. Clear? Clear on types of lipids? What types we have? Simple lipids, conjugated lipids, derived lipids. Under simple lipids we have learnt on neutral fats, which has fatty acid and glycerol. Okay. If two fatty acids are adjoined with a glycerol, it is diglyceride. Three fatty acids, triglyceride. And then fatty acids also we have learned two types, saturated and unsaturated. Saturated will have single bonds between carbon atoms. Examples include stearic acid, palmitic acid. Unsaturated fatty acids examples include oleic acid, linoleic acid, right? And then based on melting point, you have different melting points to melt, right? For example, fats will have high melting point and they remain as solids at room temperature. Ghee if we take, and right now though if you keep outside also it will be in a very solid form. And it has what? High melting point. Whereas the oils we use like sunflower oil and all, the oils which we use for regular cooking and all, it has low melting point and it stay as liquids in the room temperature, right? And then derived lipids, for example steroids, they don't have, it do not contain any fatty acid, it just includes what? Some fat like properties. The most common is what? Cholesterol, which is abundantly present in the animal tissues, that's all you can take. Okay. And it is synthesized in liver. Clear? With simple lipids and derived lipids. Then to learn on conjugated lipids, this table is enough. Okay. Remember, first type is what? Phospholipids. What are the components present here? You see, glycerol, it has alcohol, two fatty acids and a phosphate group. This is addressed as what? Choline, which has glycerol or any alcohol, fatty acids, phosphate group. Combinedly you will call it as what? Choline, which is nitrogen containing lipid, right? So they are present, or the functions not required for you. This is an additional information. If you are enthusiastic to know about this, you learn. If not, though, just type of conjugated lipids, components in it. Example, lecithin. Lectin is a secondary metabolite. Lecithin is a conjugated lipid, which is a phospholipid, right? And in our cell membrane also, we have how much % of lipid content? Cell membrane of erythrocyte, RBC in fluid mosaic model, 40% of lipid, right? In that, maximum composition is phospholipids or phosphoglycerides. Okay, they are of maximum composition or type of lipid. Next is glycolipids. It has fatty acid, alcohol, called as what? Sphingosine, and sugar molecule. Okay, fatty acid along with glycerol, it has what? Sugar. That is why glycolipid. Synthesized where? In which organelle? Golgi bodies. Okay. Lipoproteins, lipids and proteins. Chromolipids, the name itself is saying it has what? Pigments. That's all. Keratins, vitamin A. When we are learning on pigments also, we have learnt on water soluble pigments, water insoluble pigments, right? Carotenoids, chlorophylls, xanthophylls, all these are what? Water insoluble only. Chromolipids, right? So, phospholipids, glycolipids, lipoproteins, chromolipids. This column is your option. Okay? Done? this is what given in our NCERT. Palmitic acid, how to check? It has a carboxyl group, fatty acid. How many carbons along with carboxyl group? 1 plus 14, 15 plus 1, 16. And then glycerol, it has one unit of glycerol, which is trihydroxypropane. Triglyceride, it has 3 fatty acids, 1, 2 and 3, along with glycerol unit, right? So this is triglyceride. Phospholipid or lecithin. Okay, so here you can check it out with a phosphate group and nitrogen, which is nitrogen containing what? Choline. And it also has a phosphate group, lecithin, right, which is under phospholipid. And cholesterol, it exhibits fat-like properties. You cannot see a fatty acid here, right? Clear? And cholesterol is mainly responsible for maintaining the fluidity of the membrane, right? So this is what you have to be thorough with from NCERT. Clear on this? Need not to draw the structures, need not to remember everything in particular, but you have to be in a position to identify. Next, we will start with metabolism, enzymes and all, okay. So, till biomacromolecules, if you have any doubts, you ask me, I will give you 5 minutes of time. Biomacromolecules, where we have learned carbohydrates, proteins, lipids, nucleic acids, right? I hope you got at least 80% of idea, okay. You revise it, 5 minutes you just scroll on it. If you are having any doubts, you can ask me. Listen. So yes, in our living system or the living body, there are hundreds of metabolites that are present, some hundreds or thousands of metabolites are present, and no metabolite is constant all the time. It is changing. Might be some is being constructed and something is being destructed. From simple to complex, complex to simple. That cycle is happening continuously, some thousands of times in a day, and every metabolite is getting altered. So every time it is changing, that is known as what? A chemical reaction, and it is catalyzed. That reaction have to happen fast, and that reaction will be catalyzed. Okay. That catalyzed reactions are catalyzed by what molecules? Enzymes. Okay. So for example, if we take the monomers of carbohydrate is what? Monosaccharides. For example, if we take starch, it is present in the chloroplast. Nucleotide is a monomer of what? Polynucleotide chain, for example, DNA observed in chromosome. Monomer is amino acid for polypeptide observed in intermediary filaments, microtubules, microfilaments and all this. Lipids, fatty acid is one monomer. Triglycerides, it has 3 fatty acids. Example, it is present in adipose tissue. So here first, if you come into the living state, what is living state exactly? It is the non-equilibrium state which is inside the living organism. It is a non-equilibrium state and the steady state. I am saying, I am using both the terms, non-equilibrium and steady state. Sounds little different, right? If anything attains to equilibrium, that is the dead state. If you take any dead organism, is it in equilibrium? Yes, right, it is in equilibrium with the environment. We are in the steady state but non-equilibrium, which means just now I told you thousands of metabolites happening, thousands of reactions inside our body, which is a non-equilibrium steady state, that is known as what? Living state, right? So that like different molecules, if you see glucose, which is of what metabolite? Primary or secondary metabolite? Primary metabolite. Its concentration is around 4.2 millimole per liter to 6.1 millimole per liter. If you take some hormones, it will be nanograms per ml, very minute concentration. So like that, different molecules have different concentrations in our body, right? So living state is a non-equilibrium steady state to be able to perform the work. Likewise, we have to start on learning on enzymes. To say about the importance of this topic in this chapter, you have to pick 10 PYQs of NEET. Hardly 4 will be from enzymes. Means 40 to 45% of the chapter or the content is from what? Enzymes. Starting with the enzymes, most of the enzymes are proteinaceous in nature, accepted. But then there are few exceptions, I told you. Few of the RNA molecules can form up an enzyme. Exceptions, if you see, nucleic acids like RNA can also form what? Can also behave like enzymes. Like that, one example is ribozyme. Second example I can take is ribonuclease P. Ribonuclease P. Let us recall few of the functions of this RNA. If you look into the ribozyme, okay, ribozyme if we see, it helps mainly in the peptide bond formation. When a protein is formed, if you remember mol bio a bit, central dogma a bit, okay, from transcription at the end of translation, if this is an mRNA to which ribosomes are binded, tRNA will come, copy the codon, one amino acid is synthesized. Next, one more ribozyme, tRNA will come, one more amino acid is synthesized. Like that, an amino acid chain is formed. How 2 amino acids are connected? How they are connected here like this? By peptidyl transferase enzyme, that is being synthesized by ribozyme. Peptidyl transferase. Okay. So ultimately ribozyme is helpful for what? Formation of peptide bond. It is also helpful in viral replication. These are few functions that I can decode for ribozymes. Ribonuclease P. It has very minute functions actually. This helps, for example, tRNA, do you remember this clover leaf model? Closer to 3' end, what will be there here? One amino acid will be there, CCA chain, do you remember? Closer to this T-side U loop will be there, T-side C, which is ribosome binding site. Here it is DHU loop, okay, and this is the place where it is coding, okay. And from here what amino acids will be synthesized, forming up the chains like this. This is formed by what? Peptidyl transferase from ribozyme, right. Here at 5' end, to trim this, there are multiple functions of tRNA. One among that is what? They help in the translation, okay. For that, trimming of 5' end, ribonuclease P is required. Okay, these are the minute mol bio functions. If you remember, you remember. But till what extent you have to remember for your exam level is, almost of all the enzymes are proteinaceous in nature. Quite exceptions are there. Along with ribozyme, remember ribonuclease P as well. Ribozyme functions, peptide bond formation, helps in viral replication. And here ribonuclease P helps in trimming of 5' end for tRNA. That's it. But remembering the examples of the exceptional enzymes is important. Okay? Clear now? Till ribozyme, ribonuclease P and their functions. Peptidyl transferase you have to remember. These two functions, it is your choice. Okay? Next. Enzymes are what? Organic catalysts. Few of them, there are inorganic catalysts also which does not occur in the cells. What is the difference? So, every reaction in our body or in our living system can take place without the enzyme, but it will not happen in an accurate way, fastest way. Okay. Therefore, enzyme essentiality is 100% to run any reaction, right. So, this will catalyze or enhance the reaction rate. Likewise, as it is happening in the living system and its role is what? Catalytic in nature. Enzymes are known as biocatalysts. They are also known as organic catalysts inside the living organism. Enzymes, biocatalysts, organic catalysts. Then what are these inorganic catalysts? Inorganic catalysts are those which doesn't occur inside a living organisms. Quite a few you can see in thermophilic organisms whose temperature range can be very high. So, there is a difference between the enzymes, that is biocatalysts, organic catalysts and inorganic catalysts, which are important to be noted. So, these are the enzymes which are complex biological macromolecules of proteinaceous in nature. But these are simple, small sized molecules or some elements. These catalysts operate within the living organisms or biological systems. They are found in non-living systems, which means outside the cell. For example, we are performing any reactions or functions. Likewise, we need some inorganic catalyst to enhance the reaction rate, which is outside the living system. Each enzyme is highly specific to single biochemical reaction. It is highly specific to single biochemical reaction. A single inorganic catalyst can act on multiple different chemical reactions. So, here the basic difference is what? Specificity. These are highly specific and these are not specific. Next, enzymes lose activity at temperature which is above 40 degrees Celsius. This is due to denaturation. Means these enzymes have an optimum temperature, they have an optimum pH, organic catalyst. But then inorganic catalysts can perform their functions efficiently even at higher temperatures and pressures. Right? So, the basic difference is these are specific, they can run with different reactions as well. They lose the activity at temperatures above 40 degrees, means they have an optimum temperature, not just temperature, pH also. But here it can run even at higher temperatures. These are the basic differences between organic catalyst and inorganic catalyst, right? Are you taking down? Just take out the basic differences. And here these are proteinaceous mostly, but then these are simple small sized elements. Done all? Make it little faster. Yeah. Now, so enzymes from thermophilic organisms, we have learned biocatalysts or organic catalysts. They have around temperature optimum like above 40 degrees Celsius, they are unable to gain their functional capability. But then from the enzymes which are extracted from thermophilic organisms, they are thermally stable. They can be active even at higher temperatures around 80 to 90 degrees Celsius. They can ensure their stability, right? Which is a key feature of what enzymes? Thermophilic enzymes, right? So next, mechanism of enzyme action. How an enzyme action is performed. One example I will take. Sucrose. Yeah, sucrose is the substrate. To act on that substrate and convert it into simpler molecules, we have learned, right? Yeah, listen here. We have learned, right? Sucrose is what type of sugar? Good, disaccharide. And it has two monosaccharides. What are they? Glucose and fructose. What did we discuss? Any chemical reaction need to be catalyzed by the enzyme. So this reaction is catalyzed by sucrase. Sucrase is the enzyme which need to be acted on this substrate. According to lock and key hypothesis, or normally, first I'll explain the basic mechanism, then we will discuss in lock and key and induced fit model. So if this is the enzyme which is sucrase, we have discussed enzymes are of what? Tertiary structure mostly. They are proteinaceous in nature. In that we have 4 structures. Of that, enzyme is of what? Tertiary in structure. So when it is walled like that into tertiary structure, it has minute gaps, right? Some crevices and pockets like that. One such crevice whereinto the active site, that is called as active site, whereinto the enzyme can fit. The substrate can fit into this. Okay. Once again, enzymes are tertiary in structure, which can be walled into, like we have seen, like tertiary structure with many bondings. It will form crevices and pockets here and there. That crevice into which the substrate can bind is known as what? Active site. Likewise, in this enzyme, these are the active sites to which the substrate can fit into. Okay. Simply we can say ES complex, enzyme substrate complex, but there is whole lot of mechanism involved to convert this enzyme substrate into enzyme substrate complex, right? Once enzyme substrate complex is formed, it will turn to enzyme product complex. Means this substrate is turned to what? Products. 2 products. What are they? Glucose and fructose. To turn from ES to EP, this is a transient phenomena. This is reversible. Okay. And this state is known as what? Transition state. For which an energy is required that is known as activation energy. Okay, that activation energy is more in the absence of enzyme, it will be less in the presence of enzyme. Got the point? And at the end products are formed. Simple ra. Enzymes are tertiary in structure, where the enzymes will have the crevices because of the structure it has. To the crevice into which the substrate can fit, that is known as what? Active site. Okay, and this forms ES complex, enzyme substrate complex. This converting into enzyme product complex is known as what state? Transition state. And this is a transient phenomena, means temporary phenomena. It cannot be stable for longer time like this, and it is reversible also. Okay, so this for this transient state, for this transition state from ES to EP, energy is required. What is that energy called as? Activation energy. In the presence of enzyme, activation energy will be less, and EP complex, from EP complex products are released out, glucose and fructose. Clear till here, right? You have understood the basic mechanism, right? Now we will discuss on lock and key hypothesis. Is one mechanism, okay, proposed by Emil Fischer, yeah, wherein the lock is assumed like an enzyme and key is assumed like a substrate. The key is fit into the lock. That is what about this model, okay, lock and key hypothesis. But is it accepted in the biological systems, wherein not all the enzymes will have the exact sizes of the substrate, right? So this is more appreciated by Koshland proposed model, that is induced fit model, which is, for example, if we have purchased some set of globes for the entire class, same set of globes, it is fit to all the hands, right? So that hand glove model is proposed by what? Koshland, which is also known as induced fit model, where the hand is assumed like a substrate, globe is like enzyme, which is more accepted for the biological system when compared to what? Lock and key hypothesis, right? So here these are the basic differences to note. This is proposed by whom? Emil Fischer, and this is by Daniel Koshland. Here enzyme's active site is exactly complementary to the substrate shape in lock and key hypothesis, but here active site changes shape to fit into the substrate. Key is substrate here, which fits into the lock, that is enzyme. Hand is the substrate here, which fits into the enzyme, which is globe. Flexibility of the enzyme. Enzyme structure is rigid, it is not changing. Because of lack of flexibility only, this is not widely accepted. Okay, this is accepted for all the biological systems because the enzyme structure is what? Flexible. Specificity, it is highly specific. This is moderate to high specificity. Adaptability to substrate, this cannot adapt, but this can adapt to any kind of substrate, right? So these are the basic differences, of which what you have to remember is the scientists who have proposed this and the models, like what you are assuming like enzyme, what you are assuming like a substrate, right? And flexibility, that's all. This is the mechanism of enzyme action. Now what are the properties of enzymes? Properties of enzymes. Proteinaceous in nature. Can I say all enzymes are proteins? Keep the exceptions aside. If at all, if I keep the exceptions aside, I can say all enzymes are proteins. But are all proteins enzymes? No. It can be hormones, antibodies, receptors, it can be transporters, right? So they are proteinaceous in nature, first point. Chemical reaction. Enzymes do not initiate the chemical reaction. Initiation of the reaction will not happen by enzymes, but then catalyzing them, okay, is taken care of the enzymes without changing what? Chemical equilibrium. Okay. Next is efficiency. Efficiency is equal to turnover number. What does it mean? Ton means what? Substrate molecules converted per minute by enzyme. How many substrate molecules are converted by a single enzyme for how much time? 1 minute. is known as turnover number. Unchanged form. Enzymes remain unchanged after the reaction. Enzyme specificity. Enzymes are highly specific to the substrates. For example, maltase acts only on maltose, not sucrose. What enzyme acts on sucrose? Sucrase. Right? So, this is about the properties of enzymes. Proteinaceous you already know. Chemical equilibrium maintaining you all know it. But efficiency you take down please. Efficiency is equal to turnover number. What does TON means? And this specificity also you know. Take this example under specificity, enough. Done? Done all? And then chemical reactions. How can you look into the rate of chemical reaction? It is by number of products formed per unit time. And what is the influence of temperature? We learn it under factors affecting the enzyme activity also. The rate is doubled or decreased to half. For example, for every 10 degrees rise in the temperature, the reaction will be increased till it reaches the optimum temperature. And it is fall down when you are, it is reduced to half when you are decreasing it every 10 degrees Celsius. Increasing for every 10 degrees, it is doubling, and it gets half when you are decreasing for 10 degrees Celsius, right? So that is the influence of temperature. Now, what is the role of enzyme activity? We have discussed like it will improve the speed, efficiency of a reaction, rate of the reaction. For example, this is mandatory to remember, given in your NCERT. Carbon dioxide, water in the presence of the enzyme, carbonic anhydrase, it will form what? Carbonic acid. Let's say this enzyme is not there, then how much efficiently it is producing the product is with about 200 molecules of H2CO3 being formed in how much time? 1 hour. In single hour, 200 molecules of carbonic acid are formed. By using the enzyme carbonic anhydrase, the reaction will speed up dramatically with about 6 lakh molecules being formed for how much time? For every second. So that much catalytic capacity that enzyme has. The enzyme has accelerated the reaction rate with about how much time? 10 million times. Okay. 200 molecules per hour, 6 lakh molecules per second. This transition you just remember. Okay. Now, this graph is very important. Before dealing with transition state, activation energy and all, simple thing I have to tell you is, it is very important on what is present at x-axis and what is present at y-axis. In any graph you are learning. For example, in plant physiology, we have chromatograms for absorption spectra, action spectra. Likewise, we have n number of graphs in PGD, plant growth and development. And here also we have. So here, on wherever graph you are learning, first you have to focus on what? X-axis and y-axis. Here in this enzymatic graph, on x-axis what is given? Progress of reaction. On y-axis, potential energy. Anywhere in the reaction, in the graph of enzyme, if substrate is above and the products formed are below, it means in due course of formation from enzyme substrate to enzyme product, the energy is released in the form of heat. Okay, substrate is here and product is below than the substrate. Okay, which is what reaction? Exothermic reaction. Simply by looking into the graph you can say, if substrate is having high PE and product is having low, it means what? Heat is lost in the due course of reaction. It is what reaction? Exothermic reaction. If substrate is here somewhere, product is here, where energy is required, like it is gaining the energy, and this is what? Endothermic reaction. Endothermic reaction. Substrate will have low energy and products will have high energy, potential energy. Vice versa to this is what reaction? Exothermic reaction. Clear all? Now let us look at this reaction specifically. Substrate to product formation. Transition state is what? Converting from enzyme substrate complex to enzyme product complex. That is transition state. Energy required is what? Activation energy. The activation energy required without enzyme is more when compared to the activation energy which is required in the presence of enzyme. That's all you have to analyze from this graph. X and Y axis, exothermic endothermic reaction. What is transition state? From ES to EP, and then activation energy. High in the absence of enzyme, low in the presence of enzyme. Clear no? This one? And next, factors affecting enzyme activity. Temperature, pH and substrate concentration. Okay. So first we look at temperature. Look at the rate of reaction. It is increased. Rate of reaction on Y axis, temperature on X axis. The reaction rate is increased until it is reaching what? Optimum temperature. Once it is reaching the optimum temperature, from here the rate of reaction is decreased again, right? Yeah. pH also the same, you will get a bell shaped graph like this. It is increased till the pH, optimum pH, and then it is decreased. So with respect to temperature or pH, you are getting a bell shaped graph, whereas substrate concentration is an L shape, inverted L shape, wherein with the increase of substrate concentration, the rate of reaction increases. When it reaches to the saturation, it will be constant like this. Okay. So here if we see, pH is different for different enzymes. For all the biocatalysts, the optimum temperature is same. For approximately above 40 degrees Celsius, they will not perform. Okay. The enzymes are inactivated. Okay. But pH is different for different enzymes based on their site of action. Okay, these enzymes you have to remember. Pepsin, which digests proteins, where inside stomach, whose optimum pH is 2. It is interconnected, not out of your NCERT. Trypsin digests proteins in small intestine, whose optimum pH is 8. Location is pancreatic juice. Salivary amylase around 6 to 8. Location is saliva in mouth. Lipase digests fats 7.5 to 8 in pancreatic juice, released by pancreatic juice. Maltase 6 to 7 in small intestine. Renin, which is chymosin, which coagulates milk proteins in infants, right? 3.5 to 4 is its pH in gastric, released by gastric juice in infants. DNA polymerase, pH is 7.5 to 8 inside nucleus, operated during replication, DNA replication. Catalase, which breaks hydrogen peroxide to water and oxygen, whose pH is how much? 7. This is found in most of the living tissues. Clear? This is about what? Enzymes. Clear till here? Understood, no? Enzymes and their pH. Based on locations, you can remember this. If you want, you can take. You need just type of enzymes. You already know what is it acting on. Just have to note down the optimum pH and the location. So here we will see the substrate concentration. For example, to analyze the substrate concentration, it is very easy. For example, we have 10 enzymes. 10 enzymes have added on 1, 2, 3 up to 10. The rate of reaction is increasing as far I increase the substrate concentration. But after 10, even if I add more and more substrates, will it increase the rate of reaction, or the rate of reaction is decreasing? No, it will attain a constant state that is known as saturation state. And you have something called as Km, which is known as Michaelis-Menten constant. So this constant is about to show half of the maximum velocity reached. Km is the concentration of the substrate. For example, 10 enzymes I told you. 10 substrates I am adding 1 by 1. At 5, it is the value of what? Km, at which maximum velocity is reached. So Km will be equal to what? Vmax by 2. Right? Km is equal to Vmax by 2. Right? So till here it is enough. In our next class, okay, wherein we will be learning on enzyme inhibition, then on the cofactors, enzyme inhibition cofactors, nomenclature, classification of enzymes, and we will practice the PYQs. Right? Clear now till here everybody? Yeah.